Good morning everyone. My name is Dr.Anurag and I am assistant professor in home science department IIMT University Meerut. So our first unit discuss about food microbiology and food safety. So we discuss about what actually microbiology. Microbiology we have know about microbiology is the study of microscopic organisms like bacteria, viruses, fungi and protozoa. So it's examine their structural functions, growth and interactions. So this knowledge is the vital of advance in medicine, agriculture and industrials, environmental sciences. It can be help in a wide area. So we know about the microbiology and we compile the things about microorganisms, which microorganisms are good and which microorganisms are not good for human health, industrials, environmental, etc. So let me discuss in our second point some information about we have know about the history of microbiology. What is the history of the microbiology and from where the microbiology is born? So we know about the term of here. So here the history of the microbiology. So first earlier observation microbes were first observed in 17th century using the primitive microscopes. Here we seen the objective of primitive microscopes which are using in our earlier observation for the microbiology. And next is germ theory. In 19th century scientists like Louis Pasteur, Robert Koch established the germ theory of the disease. And in nowadays what is the advancement in the microbiology? So we know the developing the genetic, biochemistry, molecular biology have expanded the field of microbiology. So nowadays we have know about lot of things in the microbiology because we have get a new instruments, new techniques for the analysis and identify the microbes in microbiology sectors. So we go for next slide. So here we know about the microscope. In the term of microbiology was introduced by French chemist Louis Pasteur, who has demonstrated as the fermentation was caused by the growth of bacteria and yeast. So it is the first demonstration by the Louis Pasteur by yeast and bacteria is known as the father of microbiology. And whole process the microscope where we identify the microbes from by the lenses and microslides which used in the preparation of the cultural bacteria used for the identify the microbes. So those bacteria which can't seen by naked eyes, we use the microscopes and identify the bacteria. And the microorganisms we know, recently I told microorganism cannot be seen by naked eyes, only seen under the microscope. Why? Because small bacteria never be seen because by the naked eye. In our environment and in our environment surrounding, we have a lot of bacteria surrounded ours. So we not identify the naked eye, so we can use the microscope. And microorganisms are present everywhere. So we discuss about the microorganisms are present everywhere, earth including human beings, animal, plants, soil, water, food, atmosphere. Microorganism may be beneficial or harmful for the human being also. So in previously we discussed about where have some good bacteria and bad bacteria. So where with the beneficial and harmful for humans, environment, industrials, in everywhere, in the form of everywhere. Go for next. So what is the microbiology? we know about what is microbiology. Microbiology is the science that study microorganisms including bacteria, viruses, fungi, protozoa and algae, which they study in the microbiology, where the microbiologists examine their structure, functions, behavior and interaction with the human, animal and plants and the environment. So we can see the microorganisms where here found in the image forms like bacteria, virus, fungi, protozoa, algae. And in the term of, we have divided the microbiology in a two part. One is a micro and one is a biology. In term of micro, we can understood is the small object which is the unable to seen by the ordinary eyes, naked eye. So here we name, as the name indicate, the is a small small portions of any object which can't see by naked eyes. After the biology, in biology we divided the study of life and living organism and their morphology, anatomy and physiology, where we study the microbes, these parameters to study, identify the microbes. So here we define here also, actually microbes refers to microorganisms cannot seen by naked eyes but can only seen under the microscope. In the term of microbes, its general word describe a number of organisms. So microbes is a term generally they describe in the numerous number of the organisms. Now we let know about the morphology and dimensions of microorganisms. So here we know about the microbes, morphology and dimensions. So microorganisms inhabit diverse shape and size. Shape can vary in the spherical form, rod shaped type and spiral. So some microorganisms have unique shapes such as hyphase, corkscrew shaped, or certain bacteria we have found the shapes and identify the names under the microscope. So here we see in some images where we found the virus, bacteria, yeast and mold. So where we discuss about the shape and size structure of the microorganisms. Here is a virus range is a 20 to 400 nanometers, bacteria is a 0.5 to 10 micrometer, and yeast is a 5 to 100 micrometer, and 10 micrometer to 10 centimeter is a mold. Is the largest portion of the microorganism in any object, of the surface. So here we discuss the range. Range is a nanometer to millimeter. And we understand the morphology, dimension. Microorganism is a crucial classification, identification, the study of ecology, roles and instruction also. So here we know about the bacterial structure, bacterial name, and what's the shape, size. We identify the microorganisms. So now we know about the microorganisms and their characteristics. So here we discuss about microorganism characteristics and their characteristics. Here we know about the microorganisms or microbes are tiny living things in a bacteria, virus, fungi and protozoa, and algae. In their small size, they have initial from many ecological, industrial and biological process. So we let know about the what is a bacteria. Bacteria is a single celled prokaryotic organisms, and they play a crucial role in the cycling, disease and various industrial process, where use the bacteria are widespread in the environment within living organisms. So here we know about some bacteria's structure, where we found the structure with name. When we found the bacteria in form of a spherical, we found in the rod like, we found in a spiral. After that we go on Viruses. Viruses is in the nowadays we always aware about coronavirus. Is recently we aware about the coronavirus. Is also is a part of the microorganisms in the viruses form. So non-living entities consisting of genetic material DNA and RNA with coated of protein cells surrounded by protein coats. So they depend on the host cell from replication. They replicates, virus are replicates if they got her host cell and he replicates 1 to 2, 2 to 3, 3 to 4, like that we have replicates in the form of replications. So the tiny infection agent, they can only replicate inside the living cell and other organism. So virus always need a cells where inside he replicates like living cells and other organisms. So we have found some examples of viruses: HIV, hepatitis B, Ebola, COVID, rabies, influenza, rotavirus, hepatitis virus. Lot of viruses we have found in the studies. We know about viruses. So now we discuss about fungi. Everyone know aware about fungi. Fungi is a eukaryotic microorganism including yeast and molds, important for decomposition of food. Somewhere he have the lot of food bunch and waste of the things where we decompose and using the mold bacteria for decomposition of things and get a new byproduct from the microorganisms. So here we have the fungi. We have some fungi have beneficial and some has the hazardous for the human being. So fungus point some there we note where the fungi is a group of eukaryotic organisms that include yeast, mold and mushroom. Mushroom is mostly known about everyone. Everyone should be eat, but always be aware it's a mushroom or it's any kind of fungi. So they play initial role in the ecosystem like this decomposers, breaking down dead organic matters, recycling nutrients. Fungi can be found in a variety of environment from soil and water or plant to animals. So we have seen the fungi. We know eatable button mushrooms is a baker yeast for using for fermentation products in food industry. Corn smuts. Here is the mold bacteria which is found in bread. It's a oyster mushrooms is also, and here we have some fungi. Some fungi from beneficial relationship with the plant, while other can causes disease in plant, animals and human beings. Fungi reproduce those spores can be have complex life, cycling involving the sexual and asexual reproductions. So we go for the next slide. We understand more about the classification microorganism characteristics. So here is the next point is protozoa. Protozoa also is a part of microorganisms where we previously study. We have know about the detail in short form. We have shared the short form and a small brief introduction about the microorganisms is a part of protozoa. Protozoa is a single cell eukaryotics often found in aquatic environments and derive in the ecological roles. It's a beneficial role for the aquatic environments. So here we know about the protozoa, single cell eukaryotic organisms, free living or practices found in various environment, water, soil and within other organisms. Play an important role in decomposing organic matter, serve as a food source of other organisms. Some causes disease in human and animals. So always we know about the microorganism is a good or bad. In both condition we have identified what microorganisms we are intake or we are using. So is the microbiology is the main part of a human being. We identify the microbes and control the disease, so prevent the disease. So we always know about the microorganism, which of microorganisms is good or bad. Next What is the protozoa type? We previously studied the protozoa, but protozoa is a wide range of types like paramecium, amoeba, elogena. This is the structure of these 3 protozoas type we have found in images form. See, here's the difference between 3 protozoa types. So paramecium, which we know about the single cell organism with slippery like shape, moves using tiny hair like structure called cilia, and eat bacteria and other small organisms for survival. So paramecium is always for survival. They eat food from bacteria, other small organisms. So has 2 nucleic and a balloon like secretes for the blanching waters. And amoeba is a shape constantly change like a globe. Is shape always change. Here we see in the shape of the amoeba. Amoeba is always changing her shape. So move by stretching out parts of its body. Eat teeny organisms. Is also eat teeny organisms for its survival by surrounding them with its body and split into 2 or make other new amoebas. He split into 2 or to make a new amoebas. And now next is a Euglena is a single cell microorganism shaped like spindles, move using a long whip-like tail called a flagellum, and can make its food using sunlight. This protozoa takes food from sunlight. And he has an eye that helps finding the light. Next microorganism is alga. So here is next microorganism, and their characteristic is alga. Here is a photosynthetic organisms are living things that use a sunlight to make a food, and they can be really teeny like phytocoin in the ocean, or is be larger like a seaweeds. Is always in a small shape look likes, and as a larger shape in a like a seaweeds. So here are some points for the alga. Is a simple plant like organism that can be single celled or multicellular. It's always this like type organism that can be single celled or multicellular. In both form this alga is found. So we have found in various aquatic environments such as oceans, lakes and ponds. We always found these alga in our nearby ponds, oceans, lakes and etc., where we water will hold for long times. We found this type of alga on the surface, on the rock sides, on the stones. So here is a common in wide range of size, shape and color, including green, red and brown. Is always in a color form. We have changed the in a wide range and shapes. So it is play a crucial role in an ecosystem as a primary producer, converting sunlight into energy. They produce a converting sunlight into the energy through photosynthesis. Photosynthesis is the part of crucial role ecosystem as a primary procedure, converting the sunlight into the energy. So provide oxygen, food and habitat for many aquatic organisms used by human various industries, including foods, pharmaceutical and biofuels. This is the role of alga, where used in a human various help in a human various form like industries including foods, pharmaceuticals, biofuels. So here we see the difference between seagrass and alga. Is always relate each other, but not are same. So here the difference between is related to flowering land because they have roots, and in alga no roots. the alga on these rocks attached with the hold force. Hold fast. Hold fast. Here we see the on stone. Alga is growing. So always different from seagrass and alga. Alga not is a seagrass and seagrass not is a alga. So always aware or we know about the difference between seagrass and alga. Here the types of the alga. Number of types of the alga, just like see the structures, shapes, design, structure and color, form, design, various shapes like this. Here we know about alluminonites, fire alga, green alga, and golden brown alga, and diastome brown alga, red alga, and yellow green algas. So here we have know about the alga. Now we go next slide to know about branches of microbiology. Why we are studying about microbiology, and what sector microbiology covered. We have know about the microbiology covered the medicinal microbiology, environmental microbiology, industrial microbiology. So these three branches of microbiology covers a wide range of things, products, and develops research much more things about the microbiology. So here we know about the medical microbiology, focus on microbes that cause disease in humans, animals, and development treatment. Means if we have know about the actual microbes, which microbes impact the human beings' health. So in my medical microbiology, we identify the bacteria and develop a treatment for the particular disease. And now we have also find in the environmental microbiology. Here we study the role of microbes in ecosystem, bioradiation, and recycling organic materials. Means we have know about the industrial environmental microbiology. Study the ecosystem, biodiversity, and recycling the organic matters. And industrial microbiology, why it's important? It utilize microbes for the production of food, beverages, pharmaceuticals, and other commercial products. So is a very beneficial part of food microbiology in industry microbiology, where we develop a new product from good bacteria. So here the know about the branches. After that, what tools and techniques are used in microbiology? Because we know about the advancement. In earlier, there are not things where we know about the instruments, techniques, method, and nowadays we have achieved different types of AI methods, technology, tools. So we know about the microscope tool and techniques. First we know about microscope. Now with the microscope is our digital microscopes. In previous is ordinary microscopes. So here we have advanced in microscopy, including electron microscopy, have enabled the visualization of microorganism in greater details. We have image form and great details in depth form of the microbes, what the shape, size. We get a clear report, and we generate the report in the form of data sheet also, where we identify and properly know about the what the microorganisms are actually in the bacterial culture. Just like see you gram positive or gram negatives we have found in the microscope under the microscopic. And second tools is the culturing techniques. Culturing techniques, what is the culturing techniques? The ability to isolate and grow microorganisms in culture media, fundamental for studying their characteristics. So we have know about the culturing study. Here we take a portion of colonies and prepare and separate in every slide or petri dish, where we know they take the petri dish sample control onto the surface agar plate. We spread the samples evenly over surface, and in third we see the colonies are grown on the surface. Next is a molecular biology techniques, where we know about PCR, polymerase chain reactions, DNA sequencing, and genetic engineering have the realizations in the study of microbiological and genetics. Here is a part and example of advancement technology in microbiology. These are two techniques are in use microbiology. So these all details and all points are just an introduction to microbiology. What is microbiology? What is the branches microbiology? What is the tool and techniques in the microbiology? We are studied. So now we go for next. Why is importance of microbiology? Microbiology हमारे लिए यूज़फुल क्यों है? Why? So in a different form we see the importance in human health, agriculture, industry, environment. See the human health. Microbes can cause disease but also support immune functions and in the digestion. So it means some microbes are cause disease, but some are helpful in immune system function and digestion. Means proper digestions. We have know about the gut microbes, which are they are helps in digestions. So some microbes are important in human health. And in agriculture, microbes are used to improve crop yields, enhance soil fertility, and protect plant from the pests. So we identify the pests, what bacteria, what infection actually found in the crop. We identify and develop chemical things or microbes. We improve the crop, enhance the fertility. We kill the microbes. In industrial form, microorganisms are leveraged to the manufacture pharmaceutical, food products, biofuel, and other products. It's also an important part in a microorganism in food industry, because in food microbes we aware about a lot of microbes are helping in food products like fermented products we are using our daily life. And environmental, we know about the microbes plays a crucial role in nutrient cycling, bioremediations, and maintaining ecological balance system, because we are using the eating the other organisms, living cells, and maintaining the ecological balance. So this is this all parts are important in microbiology. So now we go for next. Applications of microbiology. Applications of microbiology. Here see in medicinal and food production. Why we discussing about the application in microbiology. So we here know about the developing vaccines, antibiotics, and diagnostic treatment fight the disease. Microbiology is nowadays is a backbone of the medicinal sector, because where we identify the microbes, and we know about the disease, and developing the vaccines, antibiotics, to diagnose the test to fight the disease. So it's a very beneficial application of microbiology in medicinal sector. Here is a food production. In food production, using microbe for the fermented preservation and creating a variety of food. Microbes is also helpful in the food production for a wide range of fermented food products and in a beverages sector. So it's also a crucial role of microbiology in food sector. And we have also know about the microbes which are destroying the food products. It's not as a compulsory to is beneficial, but sometimes microbes are destroying or harmful for the human being. So microbiology is good for human beings and also not good for human beings. So we go for next application is biotechnology. Biotechnology is also the part of the application of microbiology, using microbes for industrial process, genetic engineering and environmental cleanup. Here we see the systematic chart of the biotechnology, where in the form of the environmental cleanup and genetic engineering. So here is the also a application found in a research form, where we exploring the diversity and function of the microbes to expand scientific knowledge. Not with we stop in the one sector, we have found the microbiology, we are stable and not go for the things. So application, this application is also important for microbiology, because we have study in food, in agriculture, customer genomics, environmental microbiology, medicines, phytogenetics, genetic research, forensic science and other sectors we have implement the microbiology. So we go for next. What factors are affecting microbial growth? We have studied all the microbes and know about what the microorganisms, and now we know about the factors affecting microbial growth. Here some factor affecting microbial growth is nutrient availability. Initial nutrient like carbon, nitrogen and minerals are required for growth. So every cells, every human beings, everyone need what? Nutrient for stable initial nutrients like carbon, nitrogen and minerals are required for growth. So limited nutrients can slow down or stop microbial growth. If they not get a proper nutrition, they slow down or stop the microbial growth. So every single cell, every microbes, they need a food. So the microbial found the food in form of carbon, nitrogen and minerals. If they not get this nutrient ability, so they will also slow down or stop the microbial growth. And here is temperature. Temperature is a main parameter, because microbe grow best within a specific temperature range. If we extreme heat provided or cold, if provided extreme heat or cold, they can grow, slow down or kill microbial cells. So temperature is the basic parameter for the microbial growth, affecting growth. Here in moisture, and moisture is a part of crucial role for microbial metabolism and growth. Here is a lack of water. If the lack of water can greatly reduce the microbial population. If there is no moisture, it means the microbial population, growing condition reduce or microbial population as down. Next parameter is pH level. pH level is also the thrice best within a specific pH range. very acidic or very alkaline. If very acidic condition or very alkaline condition can disturb growth of the cellular process. Next factor is oxygen level. Oxygen level is microbes needs oxygen. Oxygen level needs microbes need oxygen to grow. Aerobic condition means where oxygen is present, while other growth without it, anaerobic condition. These are two forms. Oxygen concentration can affect microbial growth. If there is any concentration in oxygen, microbial will be affect growth and survival. So next point is a light. Light is a certain microbes like photosynthetics, bacteria, algae. Here is the algae. In previous we know about the algae is required the sunlight for photosynthesis, so require light for energy. From light he found the energy. So light intensity and duration can influence microbial activity. And now next is osmotic pressure. The concentration of solute and environmental can affect water balance in microbial cells. So high salt or sugar concentration can inhibit the growth. So here is also is a important parameter to know about growth of microorganisms, microbial growth affecting conditions. And here is a pressure. Next point is a pressure. Some microorganisms, especially those in deep sea environmental, thrives under high pressure. So they can change the pressure, can affecting their growth and survival. It means some of the certain points we know aware about the pressure, osmotic pressure, they are affecting. If any parameter we provided in enhanced form or we have slow down, so they affect the growth of microorganisms. So basically we have know about the if anything, अगर हम किसी को कोई चीज़ बहुत एक्सेस में देते हैं तो दैट मीन्स देयर ऑल्सो इज़ हार्मफुल, और बहुत कम में देते हैं दैट मीन्स दैट इज़ ऑल्सो हार्मफुल, बिकॉज दे नीड अ स्पेसिफिक रिक्वायरमेंट फॉर any growth. So here now we have know about the parameters of all about this. So now we go for next slide. So here see the factor where previously we discussed. Here is some types of graph. We know about the temperature. What's the temperature affecting the microorganisms. If they increases the temperature, if they decreasing the temperature, so what's the impact on the microorganisms. So here also we know about the nutrition factor. Microbe initial require like carbon, we have previously discussed about in that. So with this nutrient their growth by slower down or stop. So microbes require specific nutrient for growth including carbon, nitrogen, phosphorus, sulfur and various trace element. Deficiency in any initial nutrient can limit the growth. Here is the log. Here is the dead point, the sanitary phase for the microorganisms. So here is a affecting point is water availability. Microbes needs enough moisture. If there is no moisture, if there is a high moisture content, both are impact the microbes. So lack of water can greatly reduce the numbers. Here we seen the numbers of the growth we study in the microbes. And last we know about, if we are going study about microbiology and we are choosing the microbiology subject, what's the career in the microbiology, why we should decide the take the microbiology. We also know about the sector of the microbiology where our information, our knowledge will be helpful or not. So here is the medical microbiologist, where we diagnose the treatment, infection and disease, different types of labs and various institutions of medical things, etc. And environmental microbiologist is the main part of the nowadays we have aware about the more aware about the environment, access the microbial communities and ecosystem, which microbes are contribute for the our environmental, which regarding the pollutions, biodegradable, much more things. So aware about the environmental microbiologist. And food microbiologist. In nowadays FSI is also establishing the labs for the microbiology lab for the contamination of the food in several foods like cereals, spices. Nowadays we have found this type of product items, spices have found the growing condition of microbes which are harmful and somewhere hazardous for the human beings. So in this, my food microbiologist ensure the food safety and quality of the product. So it's also a crucial role in the industry and career also. And next is the industrial microbiologist. Here we develop the microbes based on the product and process, where we identify of the microbes and based on microbes based and develop the product process. After that we have research scientists. Research scientists is also we conduct the fundamental research on the microorganisms, because if we have got anything in any sector, after that we know about much more what actual after that we do. So in the research sector the scientists always conduct a fundamental research and enhance the technology advancement, innovation, things much more. So we all about today discuss things about what is microbiology, why is microbiology, application of microbiology. Now in a short view, once I repeat all the points. Here we know about the history of the microbiology, microscopes, where is the microbes invented, microorganism from a study, where is the microorganism present, what is the microbiology, why is the microbiology is important. So here the morphology and dimension of the microbiology and their characteristics. Why we study the microbiology characteristics? You know about the identification of the bacteria, viruses, yeast, mold, etc., where we characterize on the behalf of the characters, because if we know about the character, then we define the name of the bacteria, viruses and etc., which we study during the part of the study. So here we the different types of microorganisms we study: protozoa, algae. And here the branches of branch of the microbiology, what branches are running in the what branches in the microbiologies are running. Here is the medical microbiology, environmental microbiology, industrial microbiology, and tool and techniques. Next is also an important part of the tool and techniques. Here we study about what the tool and techniques are used in the current scenario. Here using a microscope, cultural techniques, molecular biology techniques. And what's the importance of the microbiology in the different sector? In different sector, the microbiology are helping human health, agriculture, industry, environment, etc. And also application of microbiology, where the microbiology is implemented: medicines, food production, biotechnology, research, and in last we have this study about the factor affecting the microbial growth. So where he know about the factors which are affecting the microbial growth: nutrient availability, temperature, moisture, pH level, oxygen levels, light, osmotic pressure, chemical agents. So here is in the graphical form of the microbial affecting microbial growths. we studied about the, and in the image form of scene we have know about the microbial conditions where they are in form of lack of the microbial counts or higher in the microbial counts. And is the last we know about the what's career in the microbiology, where we know about the medical microbiologists, environmental microbiologists, food microbiologists, industrial microbiologists, research scientists. So after that, next lecture is unit 2, food spoilage and preservations. So we will continue this topic in lecture. Thank you so much for listening me. Thank you for today's lecture.